Hi there. In this lecture, we are going to identify the differentially expressed genes. Data processing involves the development of custom R pipeline using the IFA package from Bioconductor. The pipeline included the function called exploratory logonucleotide array analysis and background adjustment to ensure the data compatibility and integrity. Normalization and expression measures were calculating using the robust multi-array method, that is a method that we use to normalize the data. Differential expression between the control and treatment group was conducted using the lemma package, and the differentially expressed genes were selected based on log to absolute value greater than P5. Probes lacking the corresponding platform annotation file were excluded from the analysis if they didn't match a gene sample. So that's how we can get the data processed using the R. What we need to do here, we need to download this file, which is our raw TAR file that we need. So here is all the data cells that we need, and you can just download this on your own. And after downloading, you can write the R script, and also I will give the R script that is required and analyze the code using R programming language. But there is an efficient way, there is a very efficient way that we can use to analyze this dataset on the Geo database as well. The tool name is Geo2R. So we can analyze the data by using Geo2R. So if we click on it, then we will see use the Geo2R to compare two or more groups in order to identify genes that are differentially expressed across experimental conditions. Results are given in the form of tables. So that's it. Option, we have the p-value adjustment. So we will go with the false discovery rate. We can also apply the log transmission, lemma precision weight. Force normalization, we are going to choose that yes, and lemma precision weight I'm gonna go with no. So here you can see the R script that is required to generate the result. You can also copy this R script and use it in your code to get the analysis done in your RStudio, but we can just go with defining the group. So first group we are going to go with the control, and the second group we are going with the tumor or treatment. So replicate 1, 2, 3 lung cancer, control SHRN. We are going to go with the control, and then the second one are the sample. So after defining the groups, so here you can see I have defined the group by clicking on it and then selecting these. So 3 are tumor sample, 3 are control samples. And then we are going to click on analyze, and it will analyze the data based on our instructions. So the differentially expressed genes will be processed. So the data is processing and the DEGs will be identified. So while we were talking, here we can get the results. So first thing that we need to see is we have to download the table that is provided of the top differentially expressed genes. So here you can see we have the top differentially expressed genes. But what kind of genes are upregulatory, what genes are of control, and what genes are of tumor? So that we can identify from our volcano plot. So here you can see volcano plot, explore and download. Here you can zoom in and see what genes So the blue color is control and the green color is tumor. So the blue color is obviously down regulated and the red color is upregulated gene. So now we will choose the 3 upregulatory genes. Here you can see 1, 2, 3. I'm gonna go with these 3 and 3 downregulatory genes. 1, 2, 3. 3 downregulatory genes and 3 upregulatory genes for our research. But for your own sake, you have to, in the other dataset, you can select more results. So once we have these volcano plot in our graph, how we can change this graph into a better presentation. So let's just open a presentation file, PowerPoint file. So open a blank PowerPoint file and then here we will start collecting our results. So the first results that we need is volcano plot. Volcano plot is for the differentially expressed genes visualization. So now that we have this, we will move to our volcano plot and here we will take a screenshot of it. You can download this as well, but taking a screenshot, I have set the maximality at 600 dpi, so my screenshots are much better. So here you can see I have a screenshot of our genes, but as you can see it is not a presentable form. My genes names are not highlighted. So what I'm gonna do is I'm gonna write the gene's name on the volcano plot for the better presentation. So how we can do that? I'm just gonna open Paint and then I'm gonna paste my file here and then I'm gonna make it a 2-window file. So here I have my visualization from the tumor versus. So here I have the gene sample of RAP1A. So this gene is, I'm just gonna type here. So I'm just gonna write the text here. RAP1A. So this is the RAPA1 gene. So I'm just gonna change these to time. So I'm just gonna make some changes. So the second gene is CCL20. So here we will write down another gene name CCL20 and that's done. So we need another name. So the third is CCL2. So I'm just gonna make some changes. So this is CCL2 gene, that is CCL20 gene, and this is RAPA1 gene. And now we will move to the downregulated section, control section. So this is SERP1A gene. So I'm just gonna type SERP1. Also you can carry out this analysis in your R programming language if you want to, but this is much more convenient for me. So if we just look at it again, SERP1, it is also SERP1, so we are going to neglect this and then we will move to CTKN2A. So we will go with this gene. Okay, I just CDNK2A. So we will write this gene name. So I think 5 is kind of enough. We have repa1, CCL20, CCL2. Is this CCL2 or CCL5? Oh, my bad. So I'm just gonna remove this. So I think you got the idea of how to do so. CCL5. Here we have CCL5 gene. So that's pretty much it. So now we have our untitled PNG file. So I'm just gonna save it to the downloads, and here I'm just gonna make it volcano plot 1. So we have the file size. So here in the downloads we will have our volcano plot 1 file, and then we will move this file to our presentation file. Oh, sorry for that. So we'll go to the paint, and then we just need, okay, so we don't need this whole slide. We just need our volcano plot. So we will just move on to there, and then we can export that. So what if I am going to do this? So here we have it. Here is our file. I'm just gonna save it, save as PNG, and then replace the name. After that, yes, we got the file perfectly good. So if we move to our presentation file, here we can see we have the file that has the names written on them. So we have modified the file. So here is the volcano plot that we need for our results. But in the results tab, you can see we have the mean difference plot as well. So we need that as well. So here is the repa one that was significantly upregulated. So here it is shown that, and here is the CDD2A that was significantly downregulated. If you want to, you can download this as well. So I always include these files in my papers as well. So I will suggest you will do the same, and then we can here I will adjust the height of the, yes, here you can see we have the perfectly good picture of our analysis. So it is the visualization of differentially expressed gene. On the one side we have the, you can also, on the one side we have the volcano plot that is showing the expression of genes, and on the other side it is showing the mean difference of the gene. So highly mean different gene is repa1 and the CDD and 2K. So next thing that we need to add the text in our figures. So here we will add it's an A, and here we will add that this is a B part of the figure. So A and B part. So before getting these, we also need the expression density and samples that we used for the study. So what are those called? Those are called the figures of distribution samples. So so we also require these. We can also get this from our page that we use to analyze the data. So we will open this image in new tab and the expression density image in new tab. So here we will see we have the samples that we needed. So I'm just gonna copy this and paste it here, and here you can see we have the perfectly good figure that shows us which samples we used for the analysis. And also we can see the expression density of these samples as well. So how much expression was in each sample, and that's pretty much it for getting all the information from the data preprocessing file and the data optimization. So we got the two figures. First one is the distributions of the sample and expression density. Second one is the volcano plot. Volcano plot A1 volcano plot is for the differentially expressed gene, and the second one is for the mean difference. You can also highlight the mean difference one if you want to, but highlighting the differentially expressed gene volcano plot is more than enough. So we have done two analysis. First one is we selected the dataset, we analyzed the dataset, we got the distribution of samples, and after that we have the expression density. After getting that, we have the differentially expressed genes. So now let's make a table that we need for the, I think we already have downloaded the table. So here we have downloaded a table. I will open it in our Excel sheet. So here I'm just gonna delete the spot ID column and retain the gene sample one. And we also have to keep the ID, p value, t value, b value, log fc, genomic accession number, and GI. You can download the GI tab, but you have to keep the gene sample tab. So what we need is we need to highlight the information in this. I'm gonna open my volcano plot, and here we can see we have the SEPR1A, a blue gene, and the first gene that we is seeing is the REPA1. So I'm going to highlight the top expressed genes. So a red one will be my upregulatory genes. So I am just gonna read the name of REPA1. After that we have the CCL20. Here we will see where we will get the CCL20. So these all are downregulatory genes. So SP1A was our downregulatory gene and CD2KNA was our downregulatory gene. So after getting this, we are seeing all of these are downregulatory genes. But here we will see our CCL20 gene, which is upregulatory. I'm gonna read this one. The CCL20 will be of red color, and then we need the CCL5 gene, which will be also red color. So we have our table of differentially expressed gene. How to exclude this table? So we need the first column, second column, our third one, and then we have the CCL20 gene column and CCL5 column. I'm just gonna copy this and paste it here and delete the unnecessary file. So here you can see we have the table of top differentially expressed genes. So I'm just gonna copy this, make a new Word file there, which will be the Bioinformatics Biomarkers Discovery for lung cancer. So that should be the title of our file and also the title of our paper, which will be the manuscript. So here we will see we have the title of our paper. So the title of our paper is Bioinformatics Biomarker Discovery for the lung cancer. You can also enhance this by Health Bioinformatics Research Performing Bioinformatics Data Analysis for Biomarker Discovery in Lung Cancer to find out potential genes or potential biomarkers. So this is the title of the paper, and then we need we are already making the figures in the slides as well. Here you can see we are making the figures. Here we need the table. So here is the first table that we got. So I'm just gonna paste the table here. So for pasting the table, we will just copy this table, and after copying this we will paste it here and then make it look well and make it look according to the research paper. So here you can see we have the table for the differentially expressed genes. So we will give the title of the table differentially expressed genes table. So of biomarkers in lung cancer. So obviously this is not the suitable form to write a paper. First we need the abstract, then we need the introduction of the paper, and then we need the methodology of how we performed all the analysis. After that we need the results, and then we will discuss that, and that is a discussion and conclusion. So that's pretty much it. Title, then we need the abstract, which you guys can write as well, and then we need the introduction. So introduction will be about the disease, and so this will all be covered in our how to use the write up to how we can use the analysis that we have performed to properly write the paper. Then I'm gonna overview you that as well. So for now we have done the two analysis, which is first we have the distribution of the samples, after we have the differentially expressed gene, third we have made the table of our differentially expressed genes that contains the different values, and now after that we need to perform some different analysis. So for that we have to we have identified some genes. After identifying some genes, we will retrieve the proteins, then we will detect the domains. After detecting the domains, we will find out the protein-protein interaction, and then we will represent that network. Then we will be moving on to our drug design and gene ontology section, and after that we will move to the protein ligand interaction. So that is pretty much it for this lecture. I hope you understood how to perform the data processing and differential expression gene analysis to identify the DEGs, which will also be your biomarkers. So that's pretty much it. Thank you so much for listening.